Sharks have long been characterized as swimming noses, basically these smell-seeking machines with not much else going on up there. But what if that's not the whole story? And what if the story changes depending on where the shark lives? This is a brain of a shark. It has ridges, bumps, depth, and structure, and it's not as simple as some might believe. That brain belongs to the Atlantic sharp-nosed shark, one of the most common small coastal sharks found along the US Atlantic and Gulf Coast. And what makes this shark particularly interesting for my project is its range. As shown on the map, it is found along both the Atlantic coast and within the Gulf of Mexico. So we have one species that is spread across different parts of the coastline. That gave me a way to ask whether where these sharks live is related to how their brains are organized. But to answer that, we first need to think about the brain as more than just one structure. Different regions are primarily associated with different functions. The olfactory bulb is primarily associated with smell, the mesocephalon with vision, the telecephalon with higher processing, the cerebellum with coordination and movement, the diencephalon with sensory integration, and the medulla with basic vital functions. These regions don't just work in isolation. They're interconnected and influence each other, but the proportion of each gives us a window into what the animal is investing in. Think of it as like a budget. Where you invest in your brain space reflects on what you need to survive. So if the environment changes across latitude, then the brain might reflect that. Proportion tells us priority, and priority tells us a little something about the environment. So that led me to my question: does where you live, where you fall along a latitudinal gradient, shape how your brain is built? My project asked whether the Atlantic sharp-nosed shark shows differences in brain organization across geography, especially between the Atlantic and Gulf individuals. The goal was to build a baseline for future work, not to prove the cause just yet, but to see whether a real geographic pattern exists. I worked with brain regions from 47 Atlantic sharp-nosed sharks. These specimens were originally collected as part of a larger study through collaborating labs across 6 sites within the species range. For each shark, I calculated each brain region as a proportion of total brain mass. So it's not just asking whether one shark had a bigger brain than the other, I was asking how the brain was divided. And from there, the core comparison became Atlantic sharks versus Gulf sharks. Do these two groups differ in how their brains are organized? The data set came from 6 collection sites within the species range. On the map, the sites are shown starting in North Carolina, moving south through Georgia, Florida, and then into the Gulf of Mexico. For my analysis, I grouped these sites into 2 broader regions, the Atlantic coast and then the Gulf of Mexico, and created this map to help visualize where those collection sites took place. This gave me 22 sharks from the Atlantic and 25 from the Gulf to compare. This grouping gave me the geographical comparison for my project. These are not just 2 labels on a map. They represent different parts of the species range in different environmental contexts. And that comparison allowed me to ask whether sharks from different regions differ in how their brains are organized. But before I could compare Atlantic and Gulf sharks, I had to deal with a problem. Sharks' brains change as sharks grow. So if one region had larger or more mature individuals in it, a direct comparison could accidentally confuse growth with geography. And this is shown in this plot. Blue dots are young of year, sharks that are under a year old. The green are subadults, sharks that are over a year but haven't reached sexual maturity. And red are adults, sharks that have reached sexual maturity. As they grow, the olfactory bulb gets proportionately larger. So if I just compared my 2 populations directly, any difference I found might just be a size difference in disguise, not a real latitudinal signal. And this wasn't just in the olfactory or smell region. Differences were shown in all of the regions. And you look at these brains, you'll see that the young of year are on the left, subadult in the middle, and adult on the right. The brain changes shape dramatically as the shark grows. The olfactory bulbs, those round structures at the very top, they get dramatically larger. And this is a real biological pattern I had to account for before making any comparison. I used a statistical approach called ANCOVA that holds body size constant, asking if these sharks were all the same size, would the difference between Atlantic and Gulf still be there? And the answer was yes. This figure shows the difference between Atlantic and Gulf sharks after controlling for body size. Blue is Atlantic, gold is Gulf. The dashed line at 0 represents what one would expect for a shark with that body size. So anything above 0 means that that region is larger than expected for the size, and anything below 0 means it's smaller than expected. I'm going to focus on the 3 regions where the real story starts to come through. The strongest signals were in the factory bulb, the mesencephalon, and the cerebellum as a secondary finding. So first, the olfactory bulb, primarily associated with smell. It's proportionately larger in Atlantic sharks. The blue box sits above 0, and the gold box sits just below. Atlantic sharks are investing more brain space in smell than Gulf sharks are, even at the same body size. And this was the most consistent finding across my location comparisons. It showed up every single time. Then the mesencephalon, which is primarily associated with vision and sensory processing. It's proportionately larger in Gulf sharks, and this was the strongest statistical result in my study. The gold box sits just above 0 here, showing that Gulf sharks consistently have more of their brain dedicated to visual processing than Atlantic sharks do. The cerebellum was also significant in the Atlantic versus Gulf comparison, with Gulf sharks showing greater proportional investment in this region. Since the cerebellum is associated with coordination and movement, this may point toward a movement-related difference. Because this result appeared in my comparisons at the margins of significance, I'm interpreting it more cautiously than the olfactory bulb and the mesencephalon. Now, if we look at the other 3 regions, the medulla, the diencephalon, and the telecephalon, those boxes are sitting right at 0 for both populations. There's not much of a difference, but that's not a failure, and that's actually an important finding in itself. Functions primarily associated with higher cognition and basic vital processes are conserved across the range, regardless of latitude. The core architecture of the brain is stable. It's the sensory toolkit, how these animals interact with their environment, or where their differences show up. The sensory regions may vary, but the core regions hold steady. So what does this mean? What might be driving these differences? I cannot establish causation from this data set, but I can use environmental context to help develop hypotheses. So first, we have water clarity, and this may be one of the most direct links to what I found. At Atlantic sites that were sampled in the study, the conditions were coastal and estuarine, with lower visibility, more sediment, and organic matter in the water column. In those conditions, vision has real limitations, and smell may become especially important for finding prey and navigating complex habitats. At the Gulf of Mexico site sampled in the study, water conditions were clearer with higher visibility, and in that environment, visual processing may be more useful. This lines up with the brain differences I observed, with Atlantic sharks showing greater investment in smell and the Gulf shark showing more investment in vision. Then there was temperature. When I added a sea surface temperature to the map, the regional differences becomes clearer. The Gulf of Mexico sites are in warmer waters overall, and the Atlantic sites sampled in the study experienced comparatively cooler conditions. This matters because temperature can affect development, so it gives us another possible explanation for why brain organization might differ between these regions. Prey is also a matter. If sharks are encountering different prey communities across these different regions, they may rely on different sensory tools to find and capture food. And finally, habitat structure. The Atlantic sites sampled in the study were coastal and estuarine, and while the Gulf of Mexico sites represent different habitat contexts than the Atlantic sites do. Different habitats can place different demands on movement, navigation, smell, and vision. So I'm not saying that this project proves which factor is responsible, but what I am saying is that brain data gives us a pattern, and these environmental differences give us hypotheses to test next. Remember that brain at the beginning, the one with ridges, structure, and complexity? This is what complexity may be doing. These sharks are not just swimming noses, they're animals whose brains reflect the environment in which they live. So what comes next? This is the first study of its kind, which means it opens up a lot of doors. The most immediate next step is expanding geographical sampling and sample size. We need more sites across the full range of the species, from the Atlantic coast and into the Gulf of Mexico, to strengthen the real latitudinal signal. From there, the natural question is whether these brain differences translate into behavioral differences in the wild. Do Atlantic and Gulf of Mexico sharks actually sense their environments differently? That is where behavioral studies comes in. And then finally, the bigger question is whether the pattern is unique to the Atlantic sharp-nosed shark or whether similar geographical differences appear in other shark species as well. But here's why this matters beyond just one species. In marine biology, we often study populations by counting individuals, tracking movements, or measuring catch. But we do not always ask what is happening on the inside, how these animals are actually experiencing their environments. The brain is an underused lens in marine biology, and if geography is associated with brain organization within a single species, as this study suggests, then we have a new framework for asking how these animals may respond as their environments change. The environment appears to shape the brain. The animals develop sensory tools that may reflect the world in which they live, the demands of a coastline, a habitat, a place. But now those environments are changing. Ocean temperatures are rising, coastal habitats are shifting, prey communities are moving. The conditions that may have shaped these patterns are not necessarily the same conditions that these sharks will face 20 years from now. And that is where this becomes complicated. Sharks can respond to their environments. Research suggests that brain morphology may have some degree of plasticity. And this study suggests that brain organization may reflect where an animal lives. But biological change takes time, and environmental change doesn't always wait. So the question becomes, will their brains change fast enough to keep up? And that's a question that's worth pursuing. Where a shark lives may be reflected in how its brain is built. Atlantic sharks show the strongest signal in smell-related brain investment. Gulf of Mexico sharks showed a stronger signal in vision-related brain investment with a secondary finding in coordination. And for the first time, this project gives us a foundation for asking why. In a changing ocean, that question becomes bigger than just one species or one brain region. It becomes a question of how sharks experience their world and what happens when that world begins to change. I want to take a moment to thank my wonderful Capstone Advisory Committee, my chair, Dr. Davi Kassav, and my committee members, Heidi Batchelor and Abel Gong. Thank you for your mentorship, your guidance, and your support throughout this project. And I would also like to thank additional support from Dr. Kara Yopak for providing me with this data set, Dr. Chris Moll, Samantha Murray, Hannah Tannenbaum, and the entire SIO community. And lastly, I want to thank my wonderful cohort, the MAS MBC class of 2026, my friends and family. Thank you for your unwavering support and enthusiasm. I could not have done this without you. Thank you so much.
Thank you so much. Fantastic presentation. I'm curious why you chose the Atlantic sharp-nosed shark.
Thank you for that question. Well, this was the data set that was available to me, but also this shark is not threatened, it's not endangered, and so that really provides us with a great baseline to look into its brain regions to see how they differ without worrying about population threats or other environmental factors that might be impacting it.
Hi,
Hi.
Great talk.
Thanks.
So were these brains from biopsies?
So the sharks were caught by bycatch from like a long line, or sometimes by bycatch from like the fishers. And so they were caught, and then we were able to dissect the brains and then look at them that way.
Were they also able to dissect the stomachs and see what they were eating?
No, I wasn't involved in the catching and the dissecting, so I'm not too sure, but that is a great question, and I can look into that.
Yeah, because it'd be great to know if the behavioral patterning, the niche partitions, if they're doing different feeding in different areas, then they'd be using different senses for different.
Yeah, that's a really great point. Thank you.
Hey, Maya, great talk, and just such a good job making, very scientific topic, but making it relevant and helping us understand why it matters. I had a question. How might you expand this study to better test latitudinal differences if you were to continue this work?
That's such a great question. So on the right side of the map are the sample locations that I had to check for my data set. And then on the left side of the map is the distribution range of the Atlantic sharp-nosed shark. And so the next step would be to collect samples from more sites across that full distribution range, especially in the sites that weren't represented in the original data set that I had. And then with more samples from more location, future work could test whether these brain-related differences continue to appear across latitude or if this was more a comparison between the Atlantic versus Gulf of Mexico. Thank you.
Great work, Maya. I was just curious, were the sample sizes similar between the Atlantic and the Gulf shark samples?
Like the amount, the
Yeah.
numbers? Yeah, they were. I had 25 from the Gulf and 22 from the Atlantic, which was very helpful to have similar comparisons. Yeah.
Great. Thanks so much, Maya.
Thank you.